All right, so we've introduced this idea of multiple alleles, that out in the real world, there are all these multiple alleles, the variants that can occur within a population. Now, one individual can only have two alleles in a diploid system, but the next individual over may have a completely different set of alleles. So this adds obviously some complication. It doesn't change the modes of inheritance or anything else. It simply adds a level where you have to think, oh, okay, how many alleles, what the possibility here? Because it could produce a greater number of phenotypes from the same locus, right? So let's try some example problems. I thought that might be the thing we want to do here. So here's a sort of a typical problem, kind of stripped out, very similar to where we started with the when we talked about the silkworms. And we have a dominant series. Only now what we've done is we've added one more allele so that we have 4 alleles instead of 3. But it's still a straight dominant system, right? We have a dominant form here that's black over sepia. Sepia is then dominant over cream and albino. Cream's then dominant over albino. And traditionally, you set up some crosses and you try to figure out, okay, what are the genotypes of some of the individuals involved? So, that's typically what we do in these genetics problems. In this one, we're saying, what's the genotypes of some parents? So what we're going to give you are some phenotypes of some parents. We're going to give you the phenotypes and some numbers for the progeny. So this is a bottom-up problem, right? We're going to give you C down here, and we're going to ask you to go back and tell me as much as you can about A and B. So let me see what I mean here. Okay, here's the first example. We've got 2 black individuals, okay, A and B, and this is C out here. These are the progeny that they produce. So now we're trying to figure out what the genotypes are. Once again, always write down the given first. Don't worry about trying to look at the problem that much. Just say, what do I know? Well, what I know is I've got a black times black. Well, to be black, I've got to have this big C allele, they're calling it here, in both individuals. I don't know what the other allele is, but I've got to have that. Okay? All right. Now, what else do I see? Well, I see that I've got about 22 of these, 7 of those. I'm thinking about a single locus. I've got my Mendel's Big 6 sheet for the umpteenth time right in front of me, sitting here. And I'm thinking about how can I get a 3-to-1 ratio? Because 7 over here, kind of divided into, get a pointer so we can see, 7 divided into 22 right here is about 3, right, approximately. So which one of these crosses gives me a phenotypic ratio of 3-to-1? Nothing new, right? It's a heterozygous times heterozygous cross. So if we think over here and look, we can see that. Now, the other thing we can do is we can say, well, let's go back to some basic logic. If it had been 24-to-1, okay, it doesn't matter. If you get 1 albino, that means because this is a completely recessive allele, it had to be hidden over here, right? In order to be albino, you've got to be little c, right? So if you're going to be little c, like that, it had to get 1 from one parent and 1 from the other. That's the way this genetic stuff works. Therefore, it must be over here. So in this problem, you've got 2 ways to solve it. Okay, you've got the ratio, which makes sense, fits perfectly with the Mendelian expectations, and you've just got logic. So hidden here was clearly some hidden alleles that produce albino. So most of them must be albino. Let's just try a couple more, kind of follow through with this. We've got a sepia and cream, and we get some progeny. So let's figure out as much as we can about sepia and cream. Well, we got CS and CC as at least each of the parents. We know that. Now we just got to figure out what the other allele is, right? Again, look what we end up with. We end up with an albino here, right? Okay, we end up with an albino, which means we've got to be hiding a little c here and a little c here. So just from that, we know that we've got, again, a heterozygous cross. Heterozygote times heterozygote, but with different dominant alleles on each side. Now black times albino. This one's going to be really easy, right? Because black, we're going to know we've got a big C. Albino, we've got 2 little c's. So in this case, we're actually going to have 3 alleles that we already know. And all we're trying to do is figure out the missing allele here. Well, it turns out we've got cream showing up. Well, cream couldn't be over here, right? Because we're already taking care of both of these alleles. Plus, if it were there, this would be cream because it dominates over albino. So this blank allele must be hiding cream. It must be a carrier for cream. So the answer there must be that we've got this basic form, okay? And just, one more to try it out. What about black times cream. Well, same thing. We've got a black times a cream here, right? We know that we've got that. We produce some sepia. Well, where could sepia be, right? It's dominant to cream and albino, so it's not neither one of those. It's recessive to black, so the only place it can hide is to be carried by black. So this allele right here must be where the sepia is coming from, okay? And there it is, okay? Now, this particular case, one of the reasons it's important to look at it is I don't know what this allele is over here for sure, right? Why? Because it could be cream. This could be a homozygous cream, and it would give this answer. It could be a heterozygote with albino, right? Because that would still, you can just try it. Put a little CC in here and make the cross. Put just a little C there and make the cross, and you should end up with these same outcomes that you have over here. Okay, so you don't know. This case, it would be legitimate to either leave a blank here and say that. The best answer, however, would be to write down both possible genotypes. Say it could be CC, it could be C, little c, okay? So either way, one of those would work. All right, let's try another one. Let's try one where we go back to that ABO blood group idea. Remember that A and O and all that are phenotypes, right? So they have an underlying set of genotypes that can produce them depending on whether they're dominant or codominant. All right, so the first question is, can an O child come from two A parents? All right, so remember these are phenotypes. So could an O child phenotype, which means he's OO, come from a parent who's blood type A? Well, the answer is yes, they could, because if both of the parents here are AO, right? They both could give an O, that would be required. And so it is possible, we don't know that it came from there, but that's what, we're working on the problem. Can you get an O child from a parent who's A and one from B? Same logic. You have to have two O's here. So this one must carry an O and that one must carry O. Is that possible to be that phenotype, B, and have an O there? Well, certainly it is. So yes, you could have an O child showing up from some parents who are A, O, and BO. How about an AB child from one parent is type A and one's O? Well, the A can be passed down, but wait, where'd the B come from? Right? That doesn't work too well. This parent's genotype here must be OO. So the only thing it can give is O. So there's no way that you can get an AB child from one parent being O and the other being A. Right? So no, it's not possible. And it's not possible because this parent has a genotype that wouldn't allow it. Okay. And then we have if an A child from an AB parent and a B parent. Okay, that takes a little thought. Can we get that? Well, there's the A. Well, you're thinking, well, this one could push down a B, so why isn't this AB? So maybe no. Well, yeah, remember, this is phenotype. And the way to be B is to either be BB, homozygous, or heterozygous BO. So in this case, yes, you could, provided that this parent right here is actually BO, you should get just a pure A child out of it. You can play with some others of these, very straightforward. But let's now take that and ramp it up one little level and kind of make it a little more complicated and talk about how it's actually kind of been used in the past, okay? One of the ways that we've actually utilized these blood type data, remember there's 100 or so blood groups that you can test for antigen-antibody reaction in blood. Blood carries lots of these for protection and for other things. Historically, they were used for what's known as paternal exclusion analysis. And court cases, at least in this country, is one of the first sets of genetics that could actually be used as evidence in court cases. But because, so many people could be type A or could be type O or type B, there's not a lot of allele variation here. So it wasn't used to prove that somebody was there, but it could be used to exclude individuals. And very often what it was trying to exclude were potential fathers, potential males, because the basic idea is that the mother and child are linked, and you know which mother the child came from. So if you know the mother's genotype and you know the child's genotype, then it has to be some explanation to match to a male. So it's called paternal exclusion analysis. And so you go to court and you would present, for whatever reason, this blood type. And it could be used for not necessarily for paternity. It could also be used for criminal cases where you had some blood. You could go in and you could you could take it. And it was very much like science, right? We try to exclude things and we say, is it possible that this individual could have been either at the scene or in this case that we're going to do here, could have been the father, right? It wasn't until the 1990s in our country that when we developed new modern molecular techniques that you'll be doing some in lab where you do DNA fingerprinting and these other things where we actually look at the DNA itself, that we started to do inclusionary genetics data, right? So now you could go out, you could take a DNA sample and you could use it as inclusionary evidence. So the same way that we would use, say, fingerprints, right? Say, yeah, I found their fingerprints there. I found their DNA there. It doesn't prove that they did the thing or anything else, but it is used as inclusionary. ABO blood group data and other blood group data was only exclusionary. You could say, we did a blood sample and this person has type O blood and the blood that was at the scene is AB, so we can exclude that individual, okay? But with the new modern techniques, it's now inclusionary. And that was a big court thing back in the 1990s. All right, so let's try a little problem, okay? So we've got A, O, and AB, and then this RH thing here. Well, that's another blood group. It turns out to be very simple, but you'll see a lot, these are lots of problems. RH positive, rhesus positive, is dominant over rhesus negative. So it's a simple dominant system. You may be aware of this from these situations where you'll have a mother who's RH negative. She'll have an RH positive fetus. She's fine for the first part, but because there's leakage through the placenta, there's exchange of materials, as you expect. Over time, after that birth or during and after that birth, she develops antibodies to RH, these RH antibodies, right? Because she's RH negative. If the next child is RH positive, then she's going to have built up antibodies. So she's going to treat it almost like she's been, had a vaccine against her own baby, right? And so she will start to try to attack the baby, her body. And these were used to be called blue babies or something because they would, when they came out, they wouldn't have the ability to exchange oxygen efficiently, and they very often, even today, require blood transfusions, and there are modern techniques to deal with it. But this is RH positive, RH negative. This is one of the blood tests that people always do when they're looking at pregnancies. Right, but it's a simple system. So what we have here, we have a mother who's known, a child who we assume is from this mother and is known, and then we ask, is this male, is either one of these loci, the ABO blood group or the RH blood group, can either one of those exclude this male, right? So is it possible to produce an O child from an AB male? No, because some mom could be AO, right? We just did that problem and could give an O, but he doesn't have an O to give. Now, RH positive, absolutely, that fits. So in our case, he would be excluded based on the ABO blood group. Right? If we take the next one, we've got the same mother. We've got an O potential male, and then we've got the AB child. So we've switched these, but AB child, okay? Can he be excluded? Well, for basically the same reason, yes. He only has OO. This is his phenotype, but his genotype is OO, right? The two alleles. He have to give an O to any children he produces, and that's not possible. With RH, yeah, the positive works out. So we wouldn't be able to exclude him based on that one. Doesn't mean he's there, but we couldn't exclude him, okay? Now what about this one? Well, these are always fun, I think, okay? We've got an O mom, RH negative, same exact genotype as the dad. He's blood type O, he's RH negative, which means he's RH negative negative, right? And the child's positive an A. Well, those didn't come from mom, they had to come from dad. Dad doesn't have an A to give, and he doesn't have a positive RH, okay? So the dominant RH factor. So in this case, we would say that both of these would be exclusionary. It'd be a strong case to exclude this individual as the potential father, okay? Now what about this one? All right, we've got A RH positive, we've got an O child negative, and we've got a B RH positive dad. And first thing, well, no, there should be a B or something. No, well, think about the ones we just did in the last problem. If this is AO and this is BO, they can produce a child who is O, right? If this individual is RH positive, heterozygote, RH negative, and mom's the same, RH positive, RH negative, the negatives could go together under simple Mendelian, and you could get an RH negative child. So could we exclude this male? Absolutely not. Okay, we cannot exclude this male. Now, is this the father of this child? You know, you need other data to indicate that, and all sorts of other evidence that would be put into play, but you couldn't exclude this individual from based on these data. These individuals up here could be excluded as fathers. Okay, so this doesn't prove anything. Again, in science, we don't try to prove things, right? We try to exclude what we can, and whatever's left over, we hope is correct. All right, and just do one more, and this is kind of to remind you that we introduced, when we did codominance, the MN blood group. And so you could use M and N in here just as easily as RH or anything else. So is this possible? Okay. Could we exclude them? Well, this guy can give a B, so the B's good, as long as she's got an O to give, because remember, this has to be, she can't be AB, because that she would show up because that's the codominance. But if she's AO, she could just make the child a BO, that would work fine. She's M. Now remember, just like in the ABO, the MN blood group, when you see M, that is homozygous M, because this is a phenotype, right? And the phenotypes directly relates back to the genotype. So she's technically MM. The child is NN, and this male is MM. Well, if he's only got M's to give, and she only has, M's to give, something's wrong here. No, well, she's MN, sorry. She could give an N, but he can't. There's no way, he doesn't have an N over here to give, right? So in this particular case, I misstated what she was for a moment. We can't exclude him based on the ABO blood group, but we can definitely exclude him based on the MN blood group, because he's got MM, all right? She's MN, I'll restate that, okay? So the child could link up to her based on the N, but it can't link up to the male right here, all right? All right, let's take and put all this together, make a sort of a complex problem. So here's a classic little trihybrid cross, right? Heterozygote first locus, heterozygote second locus, heterozygote third locus times hit, right? Okay, and when these types of problems, we would ask maybe what's the probability of something, or if it were, a fruit fly, we'd say how many of 1,000 progeny are going to be something, but this is humans, so blood type, so we'll talk about children. What is the probability that the first child will have some specific phenotype? Okay, and this first is totally irrelevant. Okay, it could be the 7th child, whatever. Okay, we don't care. Every child's independent, right? So this is really just a non sequitur there. Now we're not asking, it's a very different question to ask what's the probability of the first 3 children all having the same phenotype. Okay, that's a big, complicated probability problem. We're just asking, basically, what's the chance of any child having a particular phenotype? And the phenotype we want to have is type A blood, being Rh negative, and then MN. All right, how do we figure that out? Mendel's Big Six, not going to be anything but that. Okay, all we're going to do is we're going to say, Mendel says, from his second principle of independent assortment, that I can worry about the ABO locus, then I can come back and independently worry about the RH locus, and then I can independently worry about the MN locus. I can do one at a time. That's what Mendel's second principle says. Mendel's first principle, segregation, says I can figure out it's one of these 6 crosses, okay? And I can figure out what the outcome is. So that's all we're going to do. We're going to take the AB times AB, gives us expectations of 1A to 2AB to 1B, blood type, like that, okay? The RH, because we're now dealing with dominance and recessiveness, RH het is like big A, little a, right? Gives me a 3-to-1 expected, like this. That's phenotypic, because it asks for phenotype, not genotype, right? Okay, and then we say, okay, the codominant system of MN gives me 1 to 2-to-1 outcome. All right, so next what we got to figure out is what genotype is this phenotype, right? That's what we're after, okay? And we're saying, okay, what's that phenotype? Well, turns out that of the possibilities to be type A here, there's no AO or anything, so this has got to be AA. To be Rh negative, well, that's a recessive, so it's got to be Rh negative, and MN is codominant, so it's got to be that. All right, well, so all we got to do is look up here and say, well, there's that gets associated with a 1, Rh negative gets associated with a 1, and MN gets associated with a 2, right? So guess what? We can just use the product rule and say, it's got a 2 chance, okay? 2 out of something, right? Right? We got to figure out the something. Well, turns out there's 1, 2, 3, 4. So 1 plus 2 plus 1 is 4 parts there. 3 plus 1 is 4 parts. 1 plus 2 plus 1 is 4 parts. Using the product rule again, we can multiply those out and see that totally we expect to be 64 parts, right? All right, so now we had 2 out of something. Well, the something is 64. So it's 2 64th or 1/32nd if you do the proper reduction, as we would probably ask you to do, right? So 1 out of 32 children ought to be that phenotype from that mating. That's what it's saying. That's your chance, or that's your probabilities, okay? Now, see what I did here? Yeah, I could do a big old 64 Punnett square and, try to figure all the possible gametes that can come from this and do that, but you don't need to. Let Mendel do all the work. Mendel's second principle says I can pull all those things apart and figure out what the segregation ratios are, and I just multiply them back together to get whatever answer. And so I can go in and figure out anything I need without, going through this big old Punnett square, or without solving it. I didn't look here and find out what all 64 possible genotypes are. I just figured out the one I needed. Okay, so make these simple, right? It might have asked you about what's the probability of getting a child that's AB, Rh positive, NN, right? It'd be 2 times 3 times 1 divided into 64. So it just make it simple for yourself. Okay. All right. Well, these are some straightforward problems, that involve some things, and we could use multiple alleles in other ways. Don't forget, we could go back and do the silkworms, or we could introduce multiple alleles in any model. It doesn't have to be this. As long as you can figure out the modes of inheritance for them, you should be able to use them fairly straightforward. And I'm just trying to give you an image of the real world. The real world is there's lots of different variants out there in many systems, and you have to kind of think of them. What happens when they show up together, right, in one form?